Hello students, welcome to EPG Patashala. I am Dr. Sheikh Abdul Hussain, working as scientist at Dairy Technology Division, ICR NDRI Karnal. Today we will discuss the module Preservation of Foods by Pulsar Light Technology under the paper Principles of the Food Processing and Preservation. Nowadays, people are more interested in minimally processed foods. Non-thermal technologies are gaining popularity because of people's interest in minimally processed foods. Minimally processed foods are those treated with minimal heat treatment, and these foods undergo minimal changes in their sensory as well as nutritional characteristics. Non-thermal technologies like high hydrostatic pressure, pulsar light, and ionizing radiations will serve the purpose of minimal treatment of food products. Pulsar light technology, which uses broad spectrum of light pulses from ultraviolet to infrared, is also gaining popularity because of its non-thermal nature. The objectives of this topic include: what is pulsar light technology, mechanisms involved in destruction of microorganisms by pulsar light, and application of this pulsar light technology for food processing, and merits and limitations of this pulsar light technology. Preservation of foods by pulsar light technology. Pulsar light technology is a non-thermal food processing technique which involves discharge of high voltage electric pulses up to 70 kilovolt per centimeter, which involves discharge of high voltage electric pulses into the food product placed between two electrodes for few seconds. Pulsar light technique uses light energy in concentrated form and exposes the substrate to intense short bursts of light or pulses. Pulsar light processing used mainly to inactivate surface microorganisms on foods, packaging material and equipments. Pulsar light is recognized by several names in scientific literature, which include pulsed ultraviolet light, high intensity broad spectrum pulsed light and pulsed white light. That is, these four names can be alternatively used to represent pulsed light technology. Pulsed light includes broad spectrum of wavelengths from ultraviolet to near infrared. The wavelength distribution ranges from 100 to 1100 nanometers. That is, it contains the UV spectrum, which ranges from 100 to 400 nanometers. It also contains visible light, whose range is from 400 to 700 nanometers in the repeat, whose range is from 400 to 700 nanometers. And also, pulsar light contains infrared region, which comprises of 700 to 1100 nanometer region. Pulsar light used for food processing applications typically involves 1 to 20 flashes per second at an energy density in the range of about 0.01 to 50 joule per centimeter square at the surface of food products. Pulsar light system can produce a peak power distribution as high as 35 megawatt. Because of short pulse duration and cooling period between pulses, Heating effects are negligible in pulsar light technology. Repeat, heating effects on food products are negligible in pulsar light technology. The FDA also has approved pulsar light technology for the treatment of food for surface microorganism control, with the total cumulative treatment not exceeding 12 joule per centimeter square, and the pulse duration should be no longer than 2 milliseconds. Let us learn the principle behind the operation of the pulsar light system. Pulsar light technology involves accumulation of an electromagnetic energy in a capacitor during fractions of a second, followed by its release in the form of light within a short time, that is within nanoseconds to milliseconds. This process results in amplification of power with a minimum of additional energy consumption. Thus, the amplified high radiation energy is focused on treatment area, that is on the food surface or packaging surface, in the form of pulses to achieve decontamination. Let us see how this pulsar light system works. This diagram depicts the functioning of a pulsar light system. The main components of pulsar light system include the power supply unit, which is indicated here as high voltage DC power supply, and an energy storage capacitor, followed by the pulse configuration device, which is indicated here as pulse shaping inductor and flash lamp. Energy from high voltage power supply is stored in a high power capacitor relatively for a long period, that is a fraction of second, from which it is released to a specially designed xenon lamp unit within a much shorter period of time, that is within nanoseconds or milliseconds. As the current passes through the gas chamber of the lamp unit, a short, intense burst of light is emitted, thus focused on the treatment area. The microbial inactivation repeat, the microbial inactivation efficacy of pulsar light depends on intensity and the number of pulses delivered on the treatment area. Let us learn the mechanism behind microbial inactivation by pulsar light. The lethality of pulsar light may be attributed to its rich broad spectrum of ultraviolet content, its short duration and high peak power, and the ability to regulate the pulse duration and frequency output of flash lamps. Ultraviolet portion of the pulsar light spectrum is considered as responsible for microbial inactivation by pulsar light. Mechanisms involved in microbial inactivation by pulsar light technology include photochemical mechanism, photothermal mechanism, and physical mechanisms. Photochemical mechanism. Photochemical mechanism states that the ultraviolet portion of pulsar light causes photochemical transformation of pyrimidine in the DNA of bacteria, viruses and other pathogens to form dimers. Formation of these dimers prevents DNA unzipping for replication and the organisms becomes incapable of reproduction. This leads to their death. Shorter wavelength, that is 100 to 280 nanometer UV region, is mainly responsible for this photochemical effects. Another mechanism by which pulsar light treatment kills the microorganisms is photothermal mechanism. Photothermal mechanism states that during PL treatment, if the energy exceeds 0.5 joule per centimeter square, bacteria can be ruptured by temporary overheating caused by absorption of all ultraviolet light from a flash lamp. Relatively longer wavelength UV region and to some extent infrared region of pulsed light spectrum are also responsible for photothermal effects. Repeat, are responsible for photothermal effects. Another mechanism by which pulsed light treatment kills microorganisms is physical mechanism. Physical mechanism states that PL treatment inactivates microorganisms by collapsing the cell structure, enlarging the vacuoles, increasing the cell membrane permeability, and also by depolarization of cell membranes. Let us learn what are the various factors which affect the microbial inactivation by pulsed light. Type of microorganisms will affect the pulsed light treatment on them. For example, optical properties of cells, that is degree of scattering and absorption of light of microorganisms, will influence their inactivation by pulsed light. Some microorganisms with efficient DNA repair mechanisms are resistant to pulsed light. Another factor which affect microbial inactivation by pulsed light is interaction between light and the substrate. For example, the composition of the food sample and its surface integrity will affect the absorption of the pulsed light. So, specular or diffuse reflection can occur depending on the smoothness or roughness of the surface of the material. For translucent materials, some part of the incident light interacts with the internal structure and causes multiple internal reflections, redirections, which may decrease the effect of pulsed light. Another factor which affects microbial inactivation by pulsed light is the distance of the food source from the light. As the distance of the food source from the light increases and the depth of the substrate increases, the absorption and scattering diminishes, which leading to decreased pulsed light effect. Another factor is design of pulsed light system. Proper pulsed light system design with minimal energy losses and maximum efficiency is very much necessary to achieve effective microbial inactivation. Let us learn the applications of pulsed light technology for various kinds of food products. Applications of pulsed light technology to liquid foods. Increasing the amount of solids in liquid foods will diminish the intensity of penetration of the pulsed light, that is, in aqueous solutions, the lower the transparency, the less effective the pulsed light treatment. Liquid foods with high ultraviolet absorbance must be treated as a thin layer in order to increase pulsed light effectiveness. With increase in the number of ultraviolet absorbing components in the liquid foods, the effectiveness of pulsed light decreases. The effectiveness of pulsed light in liquid medium could be increased by minimizing the sample depth, reducing distance between pulsed light source and the sample, and or increasing the treatment time. These things must be kept in mind while treating liquid foods using pulsed light. Application of pulsed light technology to solid foods. Surface topography of solid foods influences the pulsed light effects. Pulsed light treatment of solid foods with relatively simple surfaces leads to high levels of microbial inactivation, whereas on complex surfaces like meat etc., pulsed light treatment is not much effective. Part of the radiations of pulsed light spectrum may be absorbed by food constituents on rough surfaces, leading to reduction in effectiveness of pulsed light. That is, while treating solid foods using pulsed light, we must ensure that the surface should be smooth. Application of pulsed light technology to food powders. Composition and the color of the powders influence the effectiveness of pulsed light treatment. Colored powders will absorb more pulsed light and this will reduce its effectiveness. However, thermal effects occurring due to absorption of pulsed light may result in microbial reduction in case of powders. Application of pulsed light for decontaminating food processing equipment. Pulsed light treatment is highly suitable for its applicability in decontamination of the food process equipment surfaces. The type of food product in contact with the equipment surface and the time between contamination and intense pulse treatment decide the effectiveness of the microbial inactivation on food contact surfaces. Surface topography of food contact surfaces also has a complex influence on the efficacy of pulsed light treatment. Microbial inactivation decreased in the smoothest finish due to its highly hydrophobic and reflective nature that leads to cell clustering. Let us learn the applications of pulsed light technology to decontaminate packaging material. Pulsed light technology can be effectively used to decontaminate packaging material. However, lethality of pulsed light depends on the nature of packaging material used as well as the type of microorganisms present on the packaging material. For example, coming to the nature of packaging material, if the packaging material is made of paper or plastic, then the lethality of PL will be different on these packaging materials. Let us learn the applications of pulsed light technology to decontaminate packaging material. Pulsed light technology can be effectively used to decontaminate packaging material. However, lethality of pulsed light depends on the nature of packaging material used as well as the type of microorganisms present on the packaging material. For example, coming to the nature of packaging material, if the packaging material is made of paper or plastic, then the lethality of PL will be different on these packaging materials. Mitigation of allergens using pulsed light. Pulsed light treatment can be used to mitigate allergenicity associated with consumption of some foods. Research reports showed that pulsed light treatment can effectively inactivate peanut allergens, namely ARA-H1, ARA-H2 and ARA-H3. Pulsed light treatment of soy extracts has found to decrease the levels of soy allergens, namely glycinin and beta conglycinin. However, clinical studies are needed to validate such research reports of foods. However, pulsed light can be used in combination with other non-thermal processing technologies to process various food products. Manothermosonication, high hydrostatic pressure, ultrasound, irradiation, and pulsed electric field are the various non-thermal technologies which can be used in combination with pulsed light to process food products. Pulsed light in combination with thermosonication were experimented to inactivate E. coli in orange juice using naturally occurring antimicrobial substances in combination with the pulsed light treatment can provide new avenues in controlling pathogenic bacteria. Combined use of non-thermal technologies can complement and supplement each other and helps in improving safety and quality of foods. Now let us learn the nutritional and toxicological aspects of pulsed light technology. Nutritional and toxicological aspects of pulsed light technology. Generally, pulsed light technology is considered safe for processing food products. Pulsed light treatment of foods has been approved by the FDA. The wavelengths used for pulsed light are in the non-ionizing portion of the electromagnetic spectrum, and they did not cause ionization of small molecules that the formation of radioactive byproducts is not expected during pulsed light processing of foods. Reports indicate that pulsed light treatment did not cause significant changes in sensory and nutritional quality of foods. Let us learn the merits and limitations of pulsed light technology. Merits of pulsed light technology. Inactivation of microbes by pulsed light technology is a very quick process. Consumption of energy is also very less in this process. That is, the pulsed light technology is energy efficient. Moreover, no toxic chemicals or harmful residues are obtained during pulsed light treatment of food products. Nutritional and sensory qualities of the food products are minimally affected during pulsed light treatments. Limitations of pulsed light technology. Pulsed light has low penetration power, which limits its microbial inactivation efficacy. Moreover, limited control on food heating during pulsed light treatment still remains a main concern. In food powder treatment using pulsed light, the thermal effect of pulsed light results in undesirable color changes, which has to be properly addressed. Shadow effects due to surface topography of the food samples during pulsed light treatment also limits uses of pulsed light. Foods with rough or uneven surfaces, crevices, or pores are unsuitable for pulsed light treatments. Pulsed light is not an adequate technology for certain foods like cereals, grains, and spices as they are opaque. So, dear students, let us summarize this session. Pulsed light technology is a non-thermal food processing technique. Photochemical mechanism, photothermal mechanism, and physical mechanism are the various mechanisms involved in destruction of microorganisms by pulsed light. Several factors, namely type of microorganism, interaction between light and the food source, etc., will influence the destruction of microorganisms by pulsed light. Increasing the amount of solids in liquid foods will also diminish the intensity of penetration of the pulsed light. Simple or smooth surfaces will be more effectively sterilized by pulsar light treatment than the rough surfaces. So, while treating the food with pulsar light, we must ensure that the surface should be smooth. Low penetration power of PL, that is pulsar light, limits its microbial action. PL is not an adequate technology for cereals, grains, and spices as they are opaque. So, opaque substances cannot be effectively treated by this pulsar light technology. Thank you.